Greetings, everyone. Welcome back to another after the fact lesson about physiology with me, your host. I'm hoping that this will be a short and sweet episode, but nonetheless, we're going to see where it takes us. During this installment, we're going to be talking about the senses, particularly smell, taste, sight, and hearing. And we're going to start off strong with taste. So if you're looking at my trusty whiteboard, you'll be able to see this illustration, which is actually a close-up of your tongue. In your tongue, there are these different grooves. If you were to look at it under a microscope, there are all sorts of funny shapes. There are some anvil shaped, there are some button shaped, if I can use some artistic license to describe them that way. Needless to say, there are a bunch of different shapes on your tongue, these little grooves and papilla, if you will. Within these grooves, you have what you know as taste buds, and these are obviously responsible for sending signals to your brain, processing what is currently in your mouth. So they're obviously pretty important. For every taste bud in your tongue, there are about 125 receptor cells that are capable of sending signals to your brain depending on what it is you are beginning to digest. So what I have here is an illustration of the two main types of cells that we can find on the taste bud. We're going to be looking at each individual side. I think it's important to note that this is not one specific particular cell. This is just a split illustration showing two different types. I'm going to start on the right side here because these are the type of cells that are able to perceive most different tastes. There are different receptors that respond to different, quote, tasting food. And apparently, this is news to me. It's the ions, mostly in the food, that are responsible for giving it its distinctive taste. On the left here, we see sodium and hydrogen that are responsible for both salty and sour foods, respectively. And both of those enter through a designated channel. Whereas on the right hand here, we actually have a G protein coupled receptor that binds to a free floating ligand. And this ligand can be related to either sweet, bitter, or umami foods, or meaning savory. And respectively, the molecules having to do with those flavors would be glucose, obviously, for the sweet, calcium ions for bitter foods, and for the savory umami foods, it's glutamate, surprisingly. So any of those three ligands bind to the G protein coupled receptor of this type of cell, phospholipase C will be released, triggering IP3 to bind to the smooth endoplasmic reticulum that releases its stores of calcium. As you might remember, the gradient, concentration gradient of calcium is technically higher in the extracellular fluid, but there are these small pockets within the cell, within the living cell in the or in the smooth endoplasmic reticulum that actually has a very high concentration of calcium. So it's kind of a weird technicality going on that creates this unique diffusion gradient, allowing calcium to move freely throughout the cell and get bound or to bind to these terminal and vesicular snare proteins. But before I explain that, I should probably do a little more on this side. So as I said, the salty and sour tasting foods have sodium and hydrogen respectively entering through these channels at the top of the cell. We also have some voltage-gated potassium channels that activate according to the electrical gradient. And if enough of a membrane potential is reached by this buildup of positive ions within the cell, we will have a voltage-gated calcium channel open up near the bottom here, where the vesicles are. And cause the snare proteins, which is irrespective of whatever cell you're looking at. Calcium is the powerhouse essentially of muscles, of the autonomic nervous system, really anything having to do with your nervous system. Calcium is what causes snare proteins to twist against each other and launch the contents of the vesicles outside of the cell at lightning speed, like unreal speed. And obviously, the vesicles, once they've left the cell, they're received by a neuron, and that signal is propagated all the way up to the brain, telling you, oh, this food is salty or this food is sour. So that's a little bit on taste. Smell, as you may know, is related to taste, but they kind of work independently from each other. So I'm gonna swivel over here to this illustration of Nosferatu that I drew. And this is the smell of a quesadilla wafting up your nostril, up to the structure at the very top called the olfactory bulb. If you've smelled some very strong wasabi or, yeah, just horseradish, something as strong as horseradish, you might feel a burning sensation up here. That is your olfactory bulb. And it sits right on top of this thin skeletal structure called the cribriform plate. Now, this whole deal is interesting because you actually have these neurons coming through this very thin, bony plate and ending essentially inside of this little fleshy olfactory bulb. So these neurons are uh, very thin and very sensitive, and they can be severed even after a minor car accident. You might lose your sense of smell altogether due to an otherwise harmless incident. That's just something I found interesting about it. So if we were to zoom in on these olfactory neurons that make up the olfactory nerve that go up to your brain, it would be this terminal end here. If we zoom in, we get this chicken's foot that I drew. And another interesting point, not sure how relevant it is, but we have these proteins called ACE2 that have to do with smell and taste and actually have a lot to do with blood pressure. So a point that was made in class is that the COVID virus actually disables and essentially wipes out these ACE2 integral proteins. And well, for younger people, it meant nothing more than simply losing your sense of smell for a while. It became a problem for the elderly because their blood pressure became raised as a result. And that's not really a good thing when you're already having a lot of other problems. On the other side of this terminal end of the neuron here, we have yet another G protein coupled receptor, a favorite molecule or molecular structure. And when a ligand binds, like let's say whatever ion it was emanating off of the food, let's say you're smelling glucose in the air, you're in a bakery, it smells very sweet. And that ligand binds to this G protein coupled receptor in your nose. It will generate, it will use aden, for some reason it's escaping me right now. It's a sort of cyclase, adenylate cyclase that creates CAMP. The cyclase would suggest that we are creating a molecule like cyclic AMP. And as the cyclic AMP is created, it serves as a sort of power source for various channels along the length of this neuron, sending up an sphere of influence, that's the word, of the neuron as these channels are activated, such as this sodium calcium symporter is powered by cyclic AMP apparently. And to make matters even more uh, powerful, to propagate the signal that much more, we also have this voltage gated chloride channel that not only ensures that we are having all these positive charges being imported into the cell, we also have all these negative charges being exported. So you get this huge action potential being sent straight up your nose. And since it's so close to the brain, it's just separated by that thin cribriform plate. Uh, it hits pretty powerfully. I was looking it up. I was researching this earlier, and it turns out some scientists say that your sense of smell can be thousands of times more powerful than your sense of taste. And that sounds kind of hard to believe, but if you've ever plugged your nose while eating food, it can be hard to tell the difference between something like peanut butter or tomato paste. It's kind of surprising how much smell contributes to taste, simply because of how much more powerful the action potential is, how much shorter the distance is between your nose and your frontal lobe, as opposed to your tongue, which is all the way at the bottom of your skull. So those are a few things that I found interesting about smell and taste. We're going to move on to eyesight now, and I'm going to try to explain this as succinctly as possible. But this is your eye. Just for a moment, we're going to pretend we have the lens in the middle here. We have your pupil extending out. You got all the sclera and the vitreous humor on the inside. And then at the very end here, this pinprick is the retina that receives the image. These two little squares represent objects. Photons of light hit an object. This one is in the distance. This is maybe a pickup truck on the other side of the road. And here is a little piece of dust that caught some light and is being reflected into your eye, and you just happen to see it. You'll notice that the closer object has the light refracted so much more by your pupil in order to focus that image and be able to perceive it effectively. Whereas the farther out you go, the more you stretch, the less those rays of light, so to speak, need to be bent to be comprehended. You can think of this as a sort of fisheye effect, which is why when you put a phone camera, for example, right in front of your face, and you take a 0.5, as the kids say, your face ends up looking pretty crazy because of that fisheye effect. It needs to bend, sort of distort the light rays being reflected off of your face that much more in order to be able to get the full picture. So we have our lens here, and we also have something called the ciliary body, the muscles here, which I have right here. So the pupil, as we were talking about earlier, has our lens. We have our ciliary muscles, and then suspensory ligaments, sort of ligament I was trying to remember. Here's our ciliary body, which we can represent by a bunch of tiny little muscles irradiating around the iris. This is all that the iris really is. And those streaks that you see in your iris contracting and um, expanding are the suspensory ligaments represented by these squiggly lines. And their ciliary muscles kind of go behind the pupil as illustrated over here. Now there's kind of a uh, not an antagonistic, but an inverse relationship going on between the ciliary muscles and the suspensory ligaments. So as the lens expands, these As the lens expands and flattens, these suspensory ligaments are going to become bunched up simply because all the space in the iris is being taken up by the lens, by the pupil. So, in examples of there being lower light, or you're trying to see an object better, you're squinting your eyes, it's closer to your face, you're trying to refocus, that's when these suspensory ligaments are going to bunch up like an accordion and your lens is going to expand. Now, on the inverse, if you're staring at a very bright light and your eye wants to minimize the amount that is being taken in to avoid blindness, excuse me, these suspensory ligaments are going to be pulled taut. They're going to be stretched to the limit, and your lens is going to be contracted to the max. So, how does the light get received and perceived by your eye in the first place? Well, I'm glad you asked. I have a simple illustration here. As you can see, I drew pretty much everything out. This is an ear. I drew everything out beforehand, so hopefully we'll not take 45 minutes like last time. But needless to say, these are our photoreceptor cells in the back of the eye. Here we have our pigmented bodies, and following the photoreceptor cells, that would be somewhere right here, we'd have our bipolar cells, and then we would have a blood vessel, and snaking its way around it, we would have some neurons carrying action potentials for us. So photoreceptor, bipolar receptor, or rather neuron. I'm going to give you two examples, light and dark. And I'm going to use the same illustration for both, but I'm not going to draw over them. You'll just have to follow. Here's a G protein coupled receptor carrying a molecule called 11-cis retinol. What's important about 11-cis retinol is that when it is struck by a photon, that double bond about halfway through the chain of carbons is going to be broken, and then it's going to quickly reform into, most of the time, a molecule called 11-trans retinol. When that molecule is formed, the G protein coupled receptor will cause these substances that we call subunits to aggregate. And that's really all the detail you need to know for now. But one of these subunits is called the alpha subunit, which activates yet another protein called phosphodiesterase, or PDE is the way I like to abbreviate it. Phosphodiesterase, its name suggesting that it is a sort of enzyme protein. It cleaves the bond of this molecule attached to a sodium channel right here, sort of at the main body of the photoreceptor. This is CGMP, cyclic GMP. And once it is cleaved by the activated phosphodiesterase, I know I said I wasn't going to draw on this, but for the sake of learning, none of that sodium is going to be let in once the CGMP is cleaved. You can think of it as kind of like a garage door that's being held open by a power source, but as soon as you turn it off, it just kind of slams shut. And if there's no sodium coming in, there's not going to be any sphere of influence, no belong going in through this photoreceptor. And therefore, this channel over here, which is a voltage-gated calcium channel, it's not going to detect any voltage, and therefore it's not going to be activated. No calcium to cause these snare proteins whether they're vesicular or terminal at the end of the photoreceptor here, to release glutamate. Therefore, we can say in light conditions, glutamate production is stifled in the photoreceptor. And to illustrate the use of bipolar cells better, I'm just going to skip to dark conditions because I don't want to be repeating myself. Let's say that there is no photon that strikes the G protein coupled receptor. There is no ligand binding site, by the way, it's kind of blocked off. If there is no photon action happening on the 11-cis retinol, then there are no alpha subunits produced. If there are no alpha subunits released, phosphodiesterase is not activated. If phosphodiesterase is not activated, cGMP is not cleaved, and this sodium channel is active. It remains active, sort of by default, until light is detected. Therefore, when sodium is coming in, we have a sphere of influence, or giant blong, activating the voltage-gated calcium channel, causing the snare proteins that are on the terminal end to bind with the vesicular snare proteins, shooting this glutamate out, G for glutamate, and then reaching this channel called mGlu-R6. mGlu-R6 kind of works similarly to, it's analogous to our G protein coupled receptor on the photoreceptor cell. But when it is activated by glutamate, yet another alpha subunit is produced. Don't ask me if it's the same exact type of alpha subunit. There's a chance that they don't even really know. But the alpha subunit trips a calcium channel aptly named TRPM1. Now, since there's no light and glutamate is received from the photoreceptor, this calcium channel TRPM1 does allow a sphere of influence to propagate through the cell, opening yet another calcium or voltage-gated calcium channel. And you know what it does to the snare proteins. It wrings out that neurotransmitter, well, to be received by this AMPA protein at the very end on the neuron, and the signal is taken to the brain as an action potential. So hopefully that explanation helps us understand how these different fields of vision operate. So we have, quote, fields of vision in the center of the eye and in the surrounds of that center. This concept took a while for me to understand, but hopefully I'll explain it well enough. We have our photoreceptors here from the center, and the receptors surrounding them, and then we have our bipolar cell. So let's say that light photon strikes this photoreceptor in the center. As we said earlier, light stifles glutamate production from the photoreceptor, or light stifles glutamate production from the photoreceptor. Therefore, we are by default going to have a sphere of, if it is going by the mGlu-R6 pathway, a sphere of influence to be propagated through and a signal to be received by the center. We cannot have both the center and surround fields operating at once. Therefore, if this is an on-center pathway, it is also off-surround. But why did I put a question mark here? That's because it's not always mGlu-R6 that is the receiving protein on the end of the bipolar cell. It can also be AMPA, just like we saw up here on the sort of terminating neuron that leads up to the brain. If it is AMPA, we get a sort of inverse effect. We get off center on surround. Another thing that's important to remember about the way the eye operates is that these fields of vision are named after the effect that they have on the bipolar cell. So what if a photon were to enter one of the surrounding photoreceptors? Well, I'm glad you asked. There is actually a sort of intermediate that the neurotransmitter needs to go through before it can reach the bipolar cell. And all that I think is really important to remember about this is that there's kind of an inverse relationship going on. When this, when a center photoreceptor would activate the pathway, the surrounding photoreceptor would do the opposite. So let's say glutamate is not released. If glutamate is not released, then channels will be activated and action potential will be propagated through this intermediate pigmented cell. And it will release its own neurotransmitter, which I'm pretty sure is also glutamate, to AMPA, as I was talking about earlier. These surround photoreceptors activate AMPA channels on bipolar cells. Therefore, this would be on, surround, off center. Center doesn't do anything to activate because it's releasing glutamate in this case. And it's on surround because no glutamate is being released. The intermediate is releasing the neurotransmitter, and AMPA is taking it and pushing it right through. Okay, just a couple more concepts that I'm going to go through here in this last illustration, and I'll get you out of here. So now I'm going to describe how energy carried by sound waves turns into mechanical vibrations, fluid waves, chemical signals, and then action potentials. So here's your ear. Compressed air enters through the ear canal, hits the tympanic membrane or your eardrum, and then we have these three little bones called the ossicles. We have our malleus, we have our incus, and we have our stapes right here. The stapes is sort of the terminal bone of the ossicles that receives these vibrations. So we have our sound waves being converted into mechanical vibrations. And once we get to the stapes, we reach this structure called the cochlea. If you've seen an anatomical diagram or cross section of the inner ear, you might remember seeing a snail shell like structure. That's what this is. And if we were to roll this out like a carpet, it would actually form. This isn't a great representation, but it gets wider as it folds out. And these little lines that I drew along the length of this inner part of the cochlea, which is the scala media, scala vestibula, and scala tympani. In the scala media, we have these sort of piano string like structures, shorter at the beginning and longer at the end. At the longer end, we hear lower frequencies of sound. At the beginning end, where there's more tension, we hear higher frequency sounds, which explains why over time, as we age, these uh, structures in the scala media degrade sort of one by one. Those piano strings become damaged, no longer operable, and we can't hear high pitched sounds anymore. So if we were to take a cross section of the cochlea, scala vestibula, media, and tympani, we would see a tube looking something like this. So to make sense out of this whole mess, we have our stria vasculata, we have our basilar membrane, sort of at the bottom here. We have our scalas tympani, media, and vestibula, as I said before. And this is our tectorial membrane. This is what basically contains a bunch of, or rather, it's floating in this contained part of the structure full of a fluid called endolymph. Actually, there's fluid contained in each of the different scala, perilymph in the outer layers and endolymph on the inner layer of the scala media. And here our tectorial membrane is just kind of floating freely within it. Now, what happens if you were to take a tote full of water and jam it with your fist? You would see some waves appear. That's how the mechanical vibrations are transformed in fluid waves. And this tectorial membrane, sort of like a blade of grass, is getting shaken up by that sound we just heard. And before I can explain how that gets converted into an action potential, which we really care about, I want to take a closer look at this stria vascularis, I almost said vasculata. Each individual cell in this vasculata, you see a couple of cells, has various channels. Here we have our NAC pump. It's a classic sodium potassium antiporter. We have a we have our also classic potassium leakage channels. We also have a chloride leakage channel. But something that we are less familiar with is this channel protein called NKCC. That's a sodium potassium chloride channel. It's bringing all three of those ions, symporter, into the cell at once. Now you might notice that the only ion that doesn't get pumped right back out of those three is potassium. So it appears that we are bringing this very specific positively charged ion. The reason why I have an ellipses here is because there's actually multiple layers of these cells. And each of them, just like this one, is continually pumping potassium ions inward. Hmm, interesting. But why would we need potassium ions, you might ask? That's a good question. As I mentioned earlier, this blade of tectorial membrane is getting washed around in this constantly churning endolymph fluid. And as it's undulating, word of the day, it is interacting with these columnar hair cells, these little follicles connected to our tissue. I don't think I ever heard a specific name for this structure, but our basilar membrane is contained therein. And we have these little hair structures grabbing on like little arms. We have an outer layer and an inner layer grabbing onto the tectorial membrane. And as the membrane is flipping around, it's pulling on each of these little fingers. The first one bigger than the next, and the next. And what's unique about these fingers, these sort of papillae structures, is they're mechanically gated ion channels for potassium. No surprises there. So the blade of grass flips upward, it pulls on this biggest finger, which is anchored to the lid of this mechanically gated channel, and the same happens for the next. And all of a sudden, we have all of these mechanically gated channels open for potassium to flow in freely. And so we have a number of other channels happening along the length going towards the bottom, but not interacting with the bacillar membrane. We have our sodium potassium pump. As you might remember, the sodium goes out, the potassium goes in. We have potassium leakage channels. We have voltage gated calcium channels. And all of these are really contributing to a higher overall positive charge in the endolymph outside of this membrane. And we also have our, as calcium, this voltage gated calcium channel is being opened and calcium is flowing in. We have vesicles here full of neurotransmitter. And that calcium is causing their neurotransmitter to be shot into this membrane against the electrochemical gradient, or with the gradient. And those signals are being received by the neurons and going all the way up to the brain. So that is pretty much all that I think to cover besides this little protein. This is called Preston. This is essentially just a what we call a broken chloride voltage gated chloride channel, and it was seems like it was repurposed to function more as like a an accordion is the best way I can describe it. That's what allows these um, columnar hair cells to stretch and compress. All right, well, that's all that I wanted to cover for today. I hope that it wasn't as dense as the last one and that it wasn't too bad to understand. I appreciate you all listening and hope you have a good rest of your night. See ya.